Hello everyone, my name is Geman Ying in Group 3. The topic of our group on biomedical devices in healthcare is skin-attachable non-invasive electrochemical sweat sensor for continuous glucose monitoring. This is our group member and we will illustrate this topic from the following 6 aspects separately. The first part is working principle. Sweat is one of the most accessible body fluids, and its primary biological role is for thermoregulation. Conveniently, for sampling purpose, it can be found all over the body, and it has been exploited for diagnosis purpose, including glucose. The following table shows the summary of glucose concentration, and we can see that it is clearly in sweat there is a difference between the healthy patient and diabetes patient. The left picture is a patch-based wearable devices. It has an ultra-thin and stretchable design, which enables conformal contact with the skin for efficient sweat collection and high performance under physical deformation. There are a series of structured sensors, including the humidity, glucose, pH, and temperature sensors. These multi-point sensors improve the detection accuracy. In the glucose and pH sensor shows in the right top pictures, the reference and counter electrodes are designed to be packed as closely as possible to minimize the required amount of sweat. And the working electrode consists of porous gold formed by electrode deposition and enzymes. The sensors were initially based on an electrochemical approach, which used the enzyme glucose oxidase (GOX). GOX catalyzes the oxidation of glucose to gluconolocaton in the presence of oxygen, while producing H2O2 and water as byproducts. The high H2O2 reducing catalytic activity of the pore structure confirms the enhanced sensitivity.
This part is bioinstrument. Biosensors that monitor human sweat biomarkers are required to have a wide dynamic range of response, robust sensitivity, and specificity in the physiological relevant range. We use flexible nanoporous polyamide membranes as the substrate material to fabricate the zinc oxide sensor stick. Analysis volumes as low as 1 to 3 microliter are maintained through this study of biosensing in sweat buffers. Nanopores of the flexible substrate allow absorption of sweat molecules, which are carried across to the sensor surface when sweat is introduced into the membrane. Affinity biosensing is primarily achieved through the capture probes bound to the active sensing region, which interact with the biomolecule of interest. The intercalated nanopore structures of the polyamide substrate act as a biomolecule sieve, allowing only desirable biomolecules to travel to the sensor surface. We can see the peak at the 1 kilo electron volt response to the zinc air shell of zinc oxide, indicating the disposition of the film within the nanopores. An alternating current-based signal transduction technique, electrochemical impedance Spectroscopy is used to record the sensor response over a range of frequencies from 1 MHz to 1 Hz with a small alternating current excitation signal. The interaction of a buffer with a semiconducting electrode surface modulates the capacitive reactance, which is primarily driven through charge accumulation within the EDR. These dynamic changes occurring within the EDR at the electrolyte interface are derived from the imaginary impedance component characterized by EIS. Charge modulation in the EDR is affected by its debalance, which is also measure a thickness of the double layer.
Now, let's talk about physiological status due of the device. We have studied the topic of physiological status is blood glucose. Let's look at these three aspects. First of all, what is blood glucose? Obviously, it is glucose in blood. Then, what the function it is? Blood glucose must be kept at a certain level to maintain the needs of organs and tissues in the body. Last, the clinical significance. The reason of physiological increase or decrease, eating, being hungry, or the running, and so on. The reasons of pathological increase or decrease of normal glucose metabolism, diabetes, and so on. There are two subsystems in the body that regulate blood glucose. Insulin comes from island B cells. It can promote glucose to enter liver cells, muscle cells, fat cells, and other tissue cells to synthesize glucagon, reduce blood glucose, and promoting the synthesis of fat and protein. It regulates the metabolic process. Glucagon comes from island A cell. It has a strong role in promoting glucagon decomposition and gluconeogenesis, making blood glucose significantly higher. And our targeting population is people with diabetes, so we should know something about it. Diabetes is a group of metabolic diseases characterized by hyperglycemia. The reason of type 1 diabetes or autoimmune system defect, viral infection, and a genetic factor. The reasons of diabetes type 2, the ability of insulin production in patients is not completely lost, and the syndrome of type 1 diabetes, drinking more, urinating more, eating more, and C. About the type 2 diabetes, tired and fat. Diabetes affects the people's health seriously, so we need to use the device mentioned to monitor the body in real time.
Let me talk about the safety and quality assurance. First of all, for the quality assurance, although sweat is a very useful non-invasive biological fluid for glucose monitoring, the extraction of the constant and fresh sweat sampling, which is no pollution, is the most critical. To achieve this, we choose electrical stimulation to obtain the sweat. The interference in sweat is reduced to achieve the purpose of the detection. What's more, we chose the nanotexture zinc oxide as the material. Heinonen and his research group have demonstrated the stability of nanotexture zinc oxide fields in acidic and basic conditions over an 8-week period. Therefore, it can improve the stability of the instrument, reduce the interference, increase the durability, and thus improve the accuracy of blood glucose monitoring. For the safety assurance, we are using the silicon as the pressure-sensitive adhesive material to stick the measuring instrument with human skin. It has good performance and it is non-toxic, no skin irritation, no sensitization, and breathable, so it can reduce the symptoms of skin discomfort. And the property of the electrical stimulation is controlled to less than 4 voltages, and its current is about 0.2 milliampere, which can tolerate by the human body, reduce the skin fat and skin irritation. And next, we'll talk about the social economical aspect. For this part, we found the data from the International Diabetes Federation in 2013. For the age period in 20 to 79, there are 382 million people who get the diabetes. These people make up 8.33% of the total population of this age group. And in 2035, the patient expected increasing to 592 million and 10.1 percentage of the total population. And all the patients from all over the world can be the potential customers. Because of the non-invasive nature of monitoring instruments, many people without diabetes can monitor their blood sugar without pain, thereby reducing the prevalence of the diabetes. Once the diabetes prevalence is reduced, the economic burden caused by the diabetes patient can be reduced. And this graph shows the economic burden of the diabetes militaries in USA for 2002, 2007, and 2012. As you can see, people spend a lot of money on diabetes. The direct economic burden refers to direct medical expenses and non-medical expenses due to diabetes and its complications. Direct medical expenses include the drugs, hospitalization, outpatient examination, surgery, and rehabilitation expenses. Non-medical expenses refer to transportation and food generated by the medical treatment, and accommodation cost. What's more, many mainline insurance companies have introduced insurance for diabetic patients, and we can also work with the insurance company to make the product as the gift for the project. And next, let my partner talk about the suggested improvement.
There are 3 suggested improvements in this presentation. The first is stable sweat resource. Instead of passive sweat collection, we suggest an active sweat extraction with nanomaterial substrate. Shown in this slide is the working principle of active sweat secretion called iontophoretic drug delivery system. Sweat-induced drug will be delivered by charge repulsion between the 2 electrodes. Then the skin will secrete sweat through membrane to reach the sensor. Additionally, the nanopore structure of the polyamide membrane acts as a biomolecular filter, allowing only desirable molecular to travel to the sensor surface. The number 2 part is multiple detection. Environment in practical use is complex, so we introduce this multiple detection comprised with pH sensor, temperature sensor, and humidity sensor. In this case, the detection active will collaborate itself based on the data it collected. Therefore, the accuracy will be enhanced. The third part of it is broader application. According to current use of glucose monitoring devices, we notice that target people of users are diabetes patients, and we'd like to use these devices to prevent type 2 diabetes for healthy people groups. Why type 2 diabetes? Because it accounts for at least 90% of all diabetes patients, and the number of it is still rising. Furthermore, diet is considered to be a variable risk factor of type 2 diabetes, and a low-fiber diet with a high glycemic index is positively associated with a higher risk of type 2 diabetes. High glycemic index food will stimulate blood sugar to a higher level. If the level is too high, then the excessive sugar will appear. Therefore, glucose monitoring also should be used in healthy people to prevent type 2 diabetes caused by excessive sugar and obesity. That's all. Thank you.